Hello, Dr. Vitziska. I see you.
Okay, well, hi folks. Looks like a really good turnout so far. I'm John Benson. I'm the seminar committee chair. And obviously this is a virtual seminar. I would just ask everyone to please keep, during the seminar, keep the videos and audios off if possible, just to keep distractions to a minimum. And I wanted to let everyone know that next week we have our own it'll be the last seminar of the series this fall, and it's our own Stony Cooper talking about interpreting climate change for Nebraska. So something very relevant to all of us. Should be a great talk. Yeah, and without further ado, I'll hand it over to Gabby, who's going to introduce Mason. We should have a great talk today.
Thank you, John. Hello, everyone. I see a couple of you that come from Twitter, so thank you for joining us here, and so many people from SNR too. So hello, everyone. I'm going to introduce Dr. Mason Fidino, and then he's going to start with his talk. So Dr. Mason Fidino received his PhD in ecology and evolution in 2017 at the University of Illinois at Chicago. His dissertation was on urban wildlife through space and time, and you're going to see this urban wildlife theme across not just his presentation, but my introduction of him. He has numerous publications in urban ecology and quantitative ecology, and is a reviewer for multiple journals. He has a book chapter called Trends in Long-Term Urban Bird Research, which is part of a book called Ecology and Conservation of Birds in Urban Environments. It was published in 2017, right at the time that he was finishing his PhD. So I think that's very impressive. Apparently, he's a very popular speaker too, and he has been invited as a guest speaker this semester to several universities in the US, but we were the first ones to reach him out. So thank you, Mason, for accepting our invitation first. Currently, he works at the Urbana Wildlife Institute at the Lincoln Park Zoo in Chicago, and is the analytics advisor for the Urban Wildlife Information Network, UWIN, which is an international network of researchers who systematically collect data on wildlife distributions along urban gradients. Our very own Dr. Andy Little here at SNR just joined UWIN, so there will be lots of collaborations happening between SNR and UWIN in the future. So without further ado, here's Mason, and please feel free to start whenever you're ready.
Cool. Thank you so much for the invitation. Hello from Chicago. I'm sad that I can't come out and present to everyone in person. I've got a little 19-month-old daughter that I have to kind of be thinking about in terms of COVID and whatnot still. And so I'm sitting in my dining room here and presenting for all of you today. So let me go ahead and share my screen. All right, so For those of you in the audience who are urban ecologists, this is going to be no surprise, but I'm going to go ahead and just share this with everyone. We live on an urban planet. And by that, I mean that we've got more people living in urban areas today than ever before. And the world's only going to be becoming more urban, not less, basically. By 2050, there's going to be about 66% of people worldwide living in urban areas. And because of that, because basically the world's going to be becoming more urban, not less, my colleagues and I at the Urban Wildlife Institute at the Lincoln Park Zoo have become very interested in understanding how biodiversity responds to urbanization. And of course, the field of urban ecology itself is actually quite new. It's maybe only 30, 40 years old at this point. And despite it being a somewhat new field, we have found a couple generalities that I would say, at least in terms of how species sort of respond to urban intensity or the process of urbanization. One thing that we generally see is if we have some regional species pool of little colorful dots, in urban environments, quote unquote, because what is urban is kind of hard to define. What we see at the local scale, at the habitat patches that exist in that urban area, is that we tend to see fewer species living in those habitat patches. Whereas if we compare that to sort of somewhat a natural area, far away from a city, we tend to see that there are more species living in those local habitat patches. And so, why is that? Well, the first thing that sort of happens if you've got sort of this nice little forest right here and we start urbanizing it is that we start increasing habitat fragmentation. And this can, of course, have mixed results for species. Some species can positively benefit from habitat fragmentation, whereas others can negatively benefit from habitat fragmentation. One thing that's pretty universally bad across the board, though, is that as we're converting natural habitats to sort of urban landscapes, we're just reducing the amount of overall habitat that's actually available. We're reducing fragment size as well. And lots of species really don't like that. And it can be very difficult to adapt to this near permanent change on the landscape that occurs. In addition to this, we're also introducing a slew of just different sources of mortality for species, whether that be something like automobiles or introduced invasive predators like cats. But it's not, of course, all doom and gloom for the species that can actually persist in urban environments. There's also a fair bit of opportunities if you can actually adapt to them. And that's what actually makes me quite interested and intrigued about urban wildlife ecology. And these types of opportunities come in a bunch of different varieties, whether it be anthropogenic food sources, anthropogenic water sources, even anthropogenic denning sources, or like a reduction of a predator in an area so that you can actually prey species can persist. So it's really, really great that we can actually see some types of generalities in urban areas. That there's a number of agreements between urban ecological studies. We can find these types of patterns, basically. However, cities, if I'm being honest, are actually all really quite different from one another. Even if we just look at this map of the lower 48, right? We see here that cities aren't uniformly distributed across space. That's one thing that you can definitely see. But then also just the size of cities also just vary just pretty substantially. We've got a whole lot of cities that have less than 500,000 people in them, and then a few cities that have a lot of people. And when my colleagues and I at the Lincoln Park Zoo were sort of, we've been studying urban wildlife using motion triggered camera traps throughout Chicago for the last over a decade now at this point. We were, wondering to ourselves, like, we know a lot about raccoons and opossum in Chicago, for example, but like, does that actually transfer over to your raccoon in Madison, Wisconsin, to your opossum living in Seattle, Washington? And we started, having these conversations and trying to guess and predict what types of patterns may vary. And a lot of urban ecology right now is focused on things that are occurring within cities. And so when we think about something like species occupancy or abundance, we might say that, where a species is could depend on things like how big a habitat patch is, or what type of green space it actually is, whether it's a city park or a golf course or a natural area, or what types of local species interactions are happening, all those types of questions that you may typically ask if you've got a single area in which you are studying. However, if we scale up a little bit and we start asking questions about things that can be different among cities, we can actually start, guessing maybe there's just different amount of habitat that's actually overall between cities, or the infrastructure in these cities are different. People could have different cultural attitudes even towards wildlife. Take, for example, 2 cities that I've got starred here. This is Chicago, Illinois, and Indianapolis, Indiana. One really big difference between these 2 cities is that Chicago itself is maybe about half the size of Indianapolis, which is only about 3 hours away. But it's got about 3 times the population as Indianapolis does. Indianapolis is about, I think, the size of New York City, but has 1/10 the population of New York City's. It's an incredibly sprawled out city. And so species are likely going to be responding to that built environment in Indianapolis very differently than Chicago, Illinois. Not to mention, we even put different things in our hot dogs between these 2 cities. My colleagues and I have not, of course, tested this hypothesis to see if it actually causes any difference, but it's just one of those things to, look out for in the future. And when we scale up even bigger and we start thinking about differences between regions, right? It could be climate, it could be land cover. Seattle, Washington, for example, where I grew up, is very forested and very wet, and likely has a very different wildlife community and a city, and based off of infrastructure alone, than somewhere that's more dry, like Phoenix, Arizona. And because of we see these like hierarchical sort of scale differences sort of among cities potentially that wildlife communities and biodiversity could respond to, we realized that the only way we could really tease apart these types of questions that we've been getting really excited about from doing our research in Chicago for quite a while is that we need multi-city studies to actually address these questions at these different scales. And so that's what we did. We started asking around to see if anybody would want to do research with us. And that happened maybe around 2016 or so, 2017. We started very locally in the Midwest, asking partners, say in Indianapolis and Madison, Wisconsin, actually, if they would want to, investigate how we can do multi-city studies with one another. And we started what's called UWIN, which is the Urban Wildlife Information Network. And it's a systematic multi-study collaboration to understand broad patterns of urban wildlife distributions, both within and between cities. And it's really taken off. I had no idea that it would be as big as it is now. I think we're up to 35 different cities spanning 3 different continents. Which is just really exciting. We're really working at a scale that I never thought that we could potentially be working at. And so, as part of UWIN, we have a common study design across these cities. First and foremost, we are using camera traps, motion triggered trail cameras, to sample wildlife along urban to rural gradients within each city. Camera traps, for the most part, in all of these different cities are located in urban green space, so city parks, cemeteries, natural areas. Sometimes there's some private land that gets sampled as well. And we, for the most part, follow some type of transect design. Some of our partners use grids, but we keep sites about 1 kilometer away from one another at minimum. And just to show you some variation in sort of sampling designs, here are just 2 different examples. Our transects, we've got about 120 sites, I think, in Chicago. And then Adam Aller's in Manhattan, Kansas. These are some transects that they had set up when they were part of UWIN. I'm not going to go into the methodological details for this. If you're really interested in getting into the nitty gritty of how we actually do sample, I encourage you to check out this research article. It is open access. You can also, of course, go to urbanwildlifeinfo.org. We've got all of our study design explanation stuff and whatnot there. And so we got about a few partners together, about 7 different cities in 2017, and we went out and everybody collected data all at the same exact time, and we took some time, tagged all of our data. It takes the longest, of course, with camera trap studies, and we just wanted to see what general patterns we could actually observe. And so on the x-axis of this heat map here, we've got just little acronyms for different cities. What those cities are don't really matter at this point. And then you can kind of ignore the dendrograms here. I'll be explaining the patterns that we observe here. But that heat map that you actually observe on those little squares on this plot is just the hotter it is, as it becomes more purple, actually. That means that specific species on this was detected at more sites sampled within each city. If it's gray, we did not detect that species. So I see a few different patterns that actually emerge from just looking at the raw data across all these different cities. First and foremost, we've got this group of species that are actually quite common across all these different cities. And these are your quintessential urban wildlife species throughout North America. These are your raccoon, your fox squirrel, my personal favorite, the Virginia opossum, or species like the coyote, which are pretty widely distributed at this point. We've also got some species that persist across multiple cities, but are relatively rare, or at least are less common than that first group of usual suspects. These are species like the woodchuck or the American mink. And then we've got a few unique species, some city-specific species that are just popping up in a few specific spots. Like say down at the very bottom, Austin, Texas, were the only partners in this initial study where they detected ringtail. We of course don't have ringtail in Chicago, for example. And the one thing that I got most excited about for the potential with UWIN, and you'll probably see that throughout most of the research that I'm going to be sharing with all of you, it's that this common group of species, they're not just uniformly common across all these different cities. There's actually some pretty widespread variability in how common they actually are. Take, for example, the raccoon. Austin, Texas, on the very far left there, AUTX, they were only observed in about 46% of the sites that went out and sampled. Whereas if you're in Indianapolis, Indiana, right in the middle, ININ, raccoons were at every single site that they sampled. So because we're using this, very standardized study design among partners, it's very likely that the differences that we're actually observing between these cities are ecological and not methodological. And so we're giving ourselves the opportunity to be able to explore that. Why are these species so variably different among cities? And that was the first research question that at least I tried to tackle with some of the data that was coming out of the Urban Wildlife Information Network. So the question that I had was whether or not, as cities basically become more densely populated, how common they are, does their relative occupancy actually change? So if we go from city A to city C, does, say, the occupancy of raccoon, just in general, how common they are, does it actually change between these two cities? And when we think about the types of patterns that we can kind of observe, we went out to, during like a lab meeting, we got a whiteboard and we plotted some patterns out that we might expect. First and foremost, on the x-axis here, we've got the average housing density of a city, for example. On the y-axis is some type of measure of how common a species is, so their average occupancy across cities. It could be that there's literally just no relationship for some specific species, and that's okay. It could also be that we've got some species that might have a positive relationship, so they become more common in cities that actually have a greater housing density. And these species might be the ones that we personally don't like all that much. Your pest species, things like rats, things like bed bugs, etc. And of course, we could potentially be seeing negative relationships here. So in less densely populated cities, a species could theoretically be more common, and then in very densely populated cities, species could become less common. And not only that, it's all, it's one thing to put some dots onto a graph, but could we actually take this to the next level and actually develop a model that could actually allow us to predict to cities that are out of sample, essentially? So say, for example, we've got a city here that is about 750 houses per kilometer squared, and at least in this little toy example, its average occupancy should be somewhere around about 60%. So can we actually build a model that could do this for us? And then the second question that I sort of had is, it's not just about how common a species is, right? But it's also about where they live within the city. So as a city becomes more densely populated, is a species actually living in habitat patches away from people, or are they actually living in habitat patches very close to dense human populations? And does the strength or the magnitude of that relationship actually change based off of those landscape scale differences among cities? And so to do that, I developed an occupancy model. I'm not going to go into the math behind it. It's just a multi-level occupancy model that just has interactions within and among city covariates. But we estimated each species distribution as a function of one within city covariate, so something that's calculated at every single habitat patch that gets sampled, at least from these 10 UN partners, these 10 cities that we have on this map here. So yeah, that was the housing density at each site. And then we calculated 2 among city covariates. And so that was the average housing density across all sampled locations within a city. And then also we calculated the proportion of green space that's within a city to try to get at whether or not, if the city has more habitat in general, species may respond to urbanization essentially in a different way. And what we see here is that in terms of differences in average occupancy, in terms of how species, just how common they are, there's some species that don't really respond so much to that, or we failed to detect an effect. Take, for example, coyote. They only occupied about 37% of habitat patches throughout a city, regardless of how densely populated a city actually was. Once again, this is the same exact thing as my prediction figure, so I forgot to say that before. We've got the average housing density on the x-axis and the proportion of habitat patches a species occupies in a city, or their average occupancy on the y-axis. So as housing density increases in these different cities, all these little points are different average occupancy estimates for every city. And then that line that you actually see is the predicted line predicting to other cities. We do see that as cities become more densely populated, the average occupancy of raccoon goes down. Take, for example, Manhattan, Kansas. Once again, they're almost at every single habitat patch that's observed there. I think Indianapolis is the one that's actually right next to it. Whereas on the far right side of this figure, we have Chicago, Illinois, and raccoon only occupy about half the habitat patches that we sample throughout Chicago. It's not just about the average housing density in a city, though. It's also about how much habitat is within a city. At least in this paper, we had predicted that as habitat, the amount of habitat goes up, we'd probably expect species to respond more positively to urbanization and would have a greater occupancy rate. With the cottontail rabbit, we actually found the opposite pattern. And of course, we have no causal reason for this, but it's very likely that in cities that probably have more habitat, it's probably pretty good for things that like to eat cottontail rabbit. And so in cities that are that have far less habitat, cottontail rabbits may potentially be doing a better job. But of course, we'd have to look into this a little bit further. Yeah. Oh, and then 2 different cities, just to give you a little sample. So we've got Madison, Wisconsin here. The cottontail rabbit occupy about half of these habitat patches throughout the city there, whereas in Austin, Texas, it's about 0.25. And so one thing that we also saw is that it's not only about how common a species is also about where they're distributed within a city. And so I'm not going to walk through each of these figures, and I've got a couple different examples to walk you through in the slides right after this. But one thing that's really important to pick up here is that most species their responses to urbanization actually changes due to landscape scale differences among these different cities. And so let me walk through what I actually mean from these figures. In this paper, we've actually got about 7 different species, I believe, that were in this paper. And so we've actually found this relationship with 6 different species. And so with this example that I'm actually going to be walking us through, I'm going to be using Dr. Andrew Little as my colleague here. He's just started UWIN-Lincoln. I think that they're going to be sampling sometime soon, maybe next season, who knows? But Andrew, a while back, sent the potential sites they will be sampling throughout Lincoln, Nebraska to me. And so this morning, I actually went ahead and calculated the average housing density at all those different sites. And so that came out at about 510 houses per kilometer squared. And then just as comparison, the average housing density that we have in Chicago is actually maybe about twice that, about 1,200 houses per kilometer squared. So going back to those figures that I very briefly showed you, once again, on the average housing density is on the x-axis, and then the effect of site level housing density on occupancy is on the y-axis. What that means is essentially, if Andrew went out and collected a bunch of data via camera trapping throughout Lincoln and fit an occupancy model to the raccoon data, this the slope term that he would get estimated if he included housing density within the model. More often than not, if we had to bet on it, chances are it's going to be positive. We'd see that actually this species potentially has a positive relationship with housing density throughout Lincoln, Nebraska. Whereas if we scroll over to what we would potentially observe here in Chicago, Illinois, as represented by me in that little figure, we'd actually see that raccoon responds negatively to housing density. And what makes me really excited about seeing figures like this is that, you know, in previous papers, if we were doing single city studies, we would just be disagreeing with one another. Dr. Little would potentially find that this species, the raccoon, responds to urbanization or housing density in a different way than, say, Fidino et al. 2017, something like that. But this figure actually shows that we're both right, and I love that. I absolutely love that. And it's not so much that one person is correct and one person is wrong. It's just that the cities are different from one another. Just to go one more example, here we actually are agreeing with one another. This is for the coyote, except Andrew is potentially going to find that coyote respond more negatively to housing density within Lincoln, Nebraska than we would actually find throughout Chicago, Illinois. Of course, and that's one thing that makes me really excited about bringing on new partners to this network, right? Because at least for some of the species that we've done this type of modeling technique before, we can generate these types of predictions for them and actually have like quantifiable hypotheses for them to test and evaluate. And if we get it right, that's great. And if we get it wrong, it means that's great too, because we can actually try to see why we're actually missing these, like screwing up on our predictions, essentially. Thank you for letting me use your face, Andrew, on that. I know I didn't ask you, so my apologies. So what we actually observe here is that if these data were analyzed as independent single city studies, we'd infer dissimilar species-specific responses to urbanization. But when we actually work together, when we collaborate, we actually see that these disagreements are actually explained by these very large-scale differences that occur across cities. Most importantly to me, that actually indicates that generality is definitely possible when we're actually trying to make comparisons between different urban ecological studies. But for that to happen, we actually should be trying to generalize only to cities that are similar to one another. For example, cities that have similar average housing densities. So what should urban ecologists essentially do moving forward and with this information specifically? And it's something that I just do not really see that often done in papers, and it makes me very sad, is that you need to describe the broader context of urbanization within your cities so that your results can specifically be better understood relative to other studies. If your average housing density or the amount of habitat that's within your city is wildly different than other places than other studies in other cities, and you find disagreement, it's probably not because this other study is wrong or due to potentially it could be related to methodological issues, but it's just that the cities are different from one another, and the species are potentially responding differently to them because of that. If you want more information about this study, I highly encourage you to check out the EcoApps paper. It came out last year. But yeah, another thing that I'm really excited about doing with our UWIN related data is that we can actually, I don't want to say put to bed or put to rest different hypotheses that have been generated for urban environments, but we actually have a testing board basically of a whole bunch of systematically collected data across a whole bunch of different cities that we can address different ecological hypotheses with. Take, for example, the luxury effect. So that's the hypothesis that biodiversity is basically greater in wealthier parts of the city. And there's some pretty strong evidence of that relationship with plants and with birds. But then we've also got some single city studies that the results are equivocal. Some people find positive effects, some people find negative effects. There are some review papers that perhaps indicate that it may be confounded by how urban a city is even. And so, you know, we decided like maybe this is the next project we should work on. Let's focus on trying to address and quantify the presence of the luxury effect across a whole bunch of different UWIN cities. So this data comes from about 20 different cities, a whole bunch of camera trapping days, about 1,000 sites, 4,000 camera trap deployments. These are all the cities that you see down on the left here. And of course, a few different examples of the transects that we study, from Wilmington, Delaware in the Far East, all the way over to Seattle, Washington, where I grew up. The model that we used, Chris Sutherland actually named this model incorrectly. It's actually a multi-city community model for inference about geographic variation, species richness. I will be using it all the time and citing this paper repeatedly for pretty much everything I'm going to be doing in the future. It's an absolutely amazing paper if you've got data from multiple locations. I love that this work. It's a really great paper. What does this model do? It estimates site-specific species richness among cities that basically vary in community composition. It basically helps you account for the fact that some species just simply may not be present in some regions that you're studying. Take, for example, the mule deer or the Virginia opossum. It also shares information among species, so using random effects. And then it also estimates species-specific occupancy at sites as a function of urban intensity and per capita income. And we're using per capita income as a metric for evaluating the luxury effect. To quantify the luxury effect, we went ahead and grabbed the detection-corrected species richness estimate at each site, which we can estimate with our Bayesian model. And we, for each posterior draw, we simply just calculated the correlation between species richness and per capita income of each site. So what you see on the right there is an example. So we've got our posterior correlation on the x-axis, the frequency of histogram just on the y-axis. And we see here in this at least toy example that there's a positive correlation between species richness and income, which would be an indication of the luxury effect. This derived parameter can, of course, then be investigated. We can calculate quantiles with it. It's a distribution, so we can see if it overlaps 0 or not, if it's 95% credible interval does. And then we did this also for urban intensity because we think that urban intensity may potentially confound the presence of the luxury effect. So let me show you kind of what we found using this model with all this UWIN data. This is a whole lot, I guess, now that I look at this on a slide, but you really just need to focus on the left side here, on the left figure. So on the x-axis, we've got the correlation between species richness and a city's income gradient. On the y-axis, we've got all these different cities that were that contributed data to this project, and then we've got a whole bunch of just posterior distributions essentially. And so if you kind of look down, there's maybe about 9 or 10 cities that we've got pretty strong evidence of a luxury effect. The median estimate, of course, is positive for 18 of the 20 different cities, which is impressive. But yeah, we failed to, you know, if we wanted to use some type of null hypothesis significant testing route, we failed to detect an effect of the luxury effect in maybe about half the cities in this study. On the right side here are just different examples. So St. Louis up at the top has the strongest posterior correlation, and you could see that when you look at the estimated species richness as a function of median income at sites, whereas places like Iowa City or Manhattan, Kansas, we at the very bottom there, we failed to detect that effect. This is the same exact type of figure, except
Hey, Mason, we can't hear you.
sorry about that. I forgot my headset decided to die on me. It loves to do that during lectures. I'm hoping that people can hear me now. If someone in the chat could say yes, that would be great. Anyone? We're good? Okay, thank you. All right. My apologies, gremlins. I'm going to blame it on technology. So we've got occupancy estimates here on the x-axis, how common a species is. On the y-axis, we've got a bunch of different species that we included within this model. The blue dots are the average occupancy estimate among all of our different cities. And the white dots that you actually see kind of hiding behind all of those are all the species or city specific estimates for all these different species. I'm going to revisit this a little bit later at the very end, but we see that there's actually still, once again, a lot of variability that persists, and there's some species that are very rare, like the muskrat, flying squirrel, etc., and there's other species on average that are quite common, like the raccoon. And we can quantify these types of relationships between species in response to urbanization and income. The main takeaway here is that we didn't really see species-specific responses to income whatsoever across the model. Whereas we did see quite a few species-specific responses on average across cities for responses to urbanization. Those species that predominantly and uniformly responded negatively to urbanization were typically large-bodied mammals, so lots of ungulates, for example, white-tailed deer, moose, mule deer, et cetera, et cetera. So yeah, we only found the luxury effect in about half of the cities surveyed, and so we were kind of trying to wondering, maybe why is that happening? And I think it's at the end of the day, it's that per capita income is really not the best metric for us to be able to understand what's actually occurring in cities, right? Because there's a lot of variables at play that is actually going to determine socioeconomic status in a specific part of a city. Take, for example, structural racism and classism, historical patterns of like gentrification across cities even. Those can all have a really big part to play in where green space is located, where money is actually getting placed, where money is actually getting spent in terms of infrastructure, things like that. And so when we're trying to look at things like per capita income, like it's really just not quite adequate. We need to be doing a much deeper dive basically into looking at the social ecological reasons why there's variation in things like, per capita income and how species are responding to those types of variables, essentially. And there's other mechanisms that are no doubt in play here. So there's going to be legacy effects that potentially have driven the geography of development. Cities potentially just have are just built differently, right? There's rivers that run through parts of it that are not going to be existing in other cities, et cetera, et cetera. Culturally, even speaking too, there can be individual and community level value and belief systems towards wildlife that differ, and just community standards, how people decide to, decorate their yards, et cetera, et cetera. And even just looking at the traits of the species themselves across different cities and what the regional species pool looks like relative to the species that potentially persist within a city. So going back here, and this is, I just wanted to kind of close with this. Seeing this part in this figure actually probably makes me the most excited about the potential for UWIN, because we're seeing once again with these really, really common species, just how variable they are in occupancy across cities. And there's going to be just so much extra exploration that we actually get to do then as ecologists, right? I don't get to just look at this and go, yep, every species responds uniformly across all these different cities. Like for the raccoon, like Phoenix, Arizona, I think the occupancy there was like about 0.36, whereas, you know, in Indianapolis it was about 1. And so like, you know, we've got a lot of work to do still to understand why that variation occurs. And we, you know, have the data to, instead of saying like, oh, it depends, like we've got the data to actually do deep dives and explorations into like why this type of variation occurs, which to me, you know, is just very, very exciting about, you know, where UWIN is going to be hopefully taking the future of sort of urban wildlife ecology. And with that, like seriously, none of these projects could happen without like so many different collaborators that are just absolutely amazing. I really value just like all these wonderful collaborations from all these different partners. I added Nebraska here. But then also, just thank you so much for listening to my talk. You can find me on Twitter at Mason Fidino, or my lab, or our lab rather, at LPZ UWI, or even the Urban Wildlife Information Network. If you're really interested in occupancy modeling and Bayesian statistics, I encourage you to check out my website. I've got a blog there that I, you know, from time to time add new blog posts about occupancy modeling for the most part. So yeah, I'm happy to answer any questions. And sorry for that brief little quiet moment.
Yeah, folks, people can go ahead and enter any questions they have into the chat. And I think Gabby will go ahead and read them to Mason, and yeah, hopefully get some good discussion going.
Gabby's muted right now, so I see some questions popping up in the chat, and while we're potentially trying to figure that out, I can answer them. So Ken asked, how do you evaluate all the images from your cameras? So what we've developed, short answer, we still have people going through them right now. We're trying to look into using machine learning to at least remove empty photos, because that's about 80% of every single of what you get from whenever you deploy camera traps. But we've developed a front-end web application for partners to be loading data into, which has a MySQL backend and whatnot. So we basically have our own database that their partners use, so we can ensure the integrity of the data, things like that. I spend a lot of time dealing with the data end of things during my day job, essentially. Yeah.
Josue Arteaga Torres has another question. And he's asking, is the network planning to add another methods such as audio recorders and not just camera traps?
Absolutely. We're actively, I mean, we started with camera traps because that was the easiest way to start, right? You can kind of set it and forget it. It's something that we've been doing for a decade too. So we've got the experience with them. But we are definitely looking at adding different sampling techniques. We've already done a pilot this last year using audio recorders, and I had a meeting with people at BirdNet yesterday to try to talk about how we could use you know, the tools that they've developed to identify birds across different cities, things like that. And I think the really exciting part about UWIN is that we've developed a collaboration of people that are like-minded and want to, you know, answer ecological questions. And so, you know, a whole bunch of partners have started their own smaller multi-city studies, and like, you know, doing small mammal trapping, for example. And so, yeah, like it's a great resource and network for people to come up with ideas and try out different things and increase the scale at which you're actually doing your research.
H. Blanca has a question, and he or she, I'm not sure your pronouns, please forgive me, is asking, is there any potential application of the research you do? For example, how to increase biodiversity in an urban area?
Yeah, absolutely. And I think that my boss, Seth Mackley, could speak a little bit more about this, but I end up on the more statistical side of UWIN. But we're really interfacing a lot with landscape architects and city planners to really actually talk about like, how can we better collaborate with people that are actually designing cities so we can actually give wildlife a seat at the table, make predictions, actionable predictions about where species may be under different development plans, things like that. And of course, like, at the end of the day, you can try to make some type of suggestion with the study. It's kind of white tower, like if you put it in a paper and expect people to read it. But I mean, I hate to say that that's something that we do as well. But of course, everyone does that. But yes, we are actually like trying to very closely collaborate with landscape architects and the like to facilitate that type of change. We've got a paper, I think, coming out in People in Nature in a few months with some of exactly what you're asking about. So.
Dr. Powell asks, thanks, Mason. Any evidence that cities are affected by border effects, the quality type of habitat at the edges, sharp transition to agriculture versus smoother transition to surrounding forests?
That is a great question. And I'm really hoping that someone actually starts tackling like the agricultural versus forest question throughout UWIN, because I think we have the data to do that. No one's just addressed it yet, if I'm being honest. When it comes to the edge sort of effects across different cities, I really don't know. I don't know if we've really looked at that. I think one thing that's potentially happening, at least in terms of some of the responses that we're seeing, is that the cities that have a lower population density, for example, for the most part tend to just be smaller in general. And so I think that it's much easier for a species, if you're a raccoon, to sort of take a walk through Iowa City than if you were sort of a raccoon walking through Chicago, Illinois. So that's potentially one thing that I think is actually going on that we still need to evaluate.
Yes, actually, I love that question, but yeah. So Dave Widin, I think, maybe, because it says Oak Savannah. I think it's Dave Widin, right? Dr. Widin, is that you? I don't know. So he's asking, what about the quality of green space? As a plant ecologist, I might consider how far the green space deviates from pre-settlement or natural vegetation. I know Chicago has lots of prairie savanna groupies trying to restore urban forest parks to more open savanna conditions. So does that matter, do you think?
Certainly, I think that matters, but it may not matter as heavily for some of like the usual suspects that we'd pick up on a motion triggered camera trap, right? Like raccoons and opossum. But we definitely have looked at least in Chicago specifically, about how much variation there is in different types of green space. And so say, for example, like the wildlife communities that you see in cemeteries or golf courses are actually pretty comparable to what you, at least in terms of species richness, pretty comparable to what you'd actually see in a natural area. Whereas if you're in a city park, it's about a half of the species richness. Beta diversity among all those different site pairs, though, is actually quite different from one another. And so we're, a lot of what the types of research that I'm trying to do now is like test something out within Chicago for a little bit and then apply it widely to the network. And so that's one research question that's actually just like asking to be done. Someone just with the statistical know-how just needs to go for like, here's the model that was written for Chicago. Let's expand it up to what's going on across, North America, essentially.
Okay, Josue is asking another question again, and he's saying, how can we join the network as individual researchers?
The best way to do that is to go to www.urbanwildlifeinfo.org. You could, of course, email me or send me a direct message on Twitter. I can direct you to the correct spot for that. But we've got a coordinator for the network who will answer some of your initial questions, and then we basically set up some meetings with Dr. Seth Mackley and myself, and we help people work through the process of site selection if they need it, et cetera, et cetera. We're trying to be as inclusive and collaborative as possible, and I think our success rate is showing because of that. We've got a lot of different partners contributing data to this project, and it's really fun. So I'm so amazed that I get to work with so many amazing people doing this type of stuff. So yeah, that's the way I would do it. So you could reach out to me directly and I can get you in touch with the correct people too. So
Catherine Chan is asking, is there much historical data for various cities, and has much work looked into how species have adjusted to human population increase in development? Do you think multiple cities or different sizes can be used as a type of chronoconsequence method?
Ooh, there's a lot to unpack there. I won't be able to address this specifically with the data coming out of UWIN. I do know though that species will definitely be like acclimating to, at least from some of the bird review stuff I've done. That specific species can acclimate to urban environments over time. They can use green space, for example, to get sort of used to urban areas and then potentially later colonize the entire city. I think blackbirds in Europe have done that. But in terms of chrono sequencing, I don't really know. I think that in a perfect world, if you can grab all those different cities and remove all of the higher scale variation that exists, things like climate, things like the infrastructure potentially, then yes, I think you could use different size cities. But I think that there's going to be variables that differ at the same exact time that, as doing an observational study, it can be very, very difficult to control for. If I can make 10 different Chicagos for replication, that would be amazing, but we know that's physically impossible. So sorry if I didn't answer that directly.
Okay, so yeah, Catherine, if you have any follow-up questions or something, feel free to put them in the chat. Ken Chapman is asking, are the people in your domain skilled in the use of statistical tools to do these investigations? 1000% they are, but yeah. Mason will say that. Are tools like TensorFlow, CAFI, SAS, or other machine learning tools employed, or is it mostly just R? Is there general access to your databases for our own research, or is that data restricted to just UWIN?
Just R? Wow. No. R is a wonderful tool that's quite general. But yes, there's definitely people in the field that can use a whole bunch of different techniques. Like I do lots of programming in Python, and I'm having to learn JavaScript now to deal with front-end web application stuff. So yes, of course, like any human can learn how to program in different programming languages. But is there general access to our databases? No. As part of UWIN, you sign an MOU where each partner is an owner of their own data. And so we cannot just provide general access to those data. We do, however, when we publish studies, make our data openly available. And so if you look at least the cleaned data, and we do that for a couple of reasons. Like one, it does take quite a bit of work to go ahead and clean all those data and get them ready for analysis. And then two, actually that's pretty much just the one. Like it's like, at least for our first study, like it took me like 3 or 4 months just to go through and like make heads or tails of like fixing database errors, things like that. It's just a lot of work, and like I would hate to just give people general access and have them not know that there's errors that lie within. So yeah, I would say that like if you like, I've got every single data set like published on Zenodo that we've used from published research, also on GitHub, for example. So yeah, if you look at any of our publications related to UWIN, you should be able to find the data in the supplemental material, basically.
And last question from H. Blanca is another question. What international cities do you work with? Are you working with any international cities, Mason?
Yep. Yeah. I mean,
which ones?
So I can't remember all of our Canadian partners, but that is kind of cheating international, right? Because it's just another North American partner. So I think the more direct answer to your question is that we actually just had Freiburg, Germany, join the network, which is really exciting. And then we're just about to get sampling underway in South Africa. I can't remember the city off the top of my head, but I can't remember what city. But they've got urban baboon there in South Africa, which I'm really, really excited to. I don't know how well like our camera traps are going to be able to sample, or their camera traps will be able to sample them. But they also have eastern gray squirrel down in South Africa as well as an invasive species. So it will certainly be interesting to see how habitat selection sort of changes for gray squirrel out of their range, basically.
Mason, we have to talk then, because I think Guatemala City could join UWIN. It would be awesome because we have raccoons, we have opossums, gray foxes, a lot of little creatures, even margates living near cities.
Yeah. So yeah, I mean, we're actively like really like that's the one like we right at this point we feel like that a lot of like US and Canada partners, like that will take care of itself at this point in terms of people wanting to come and chat with us to try to spin up a new city, a new UWIN partnership. And so what we're trying to spend most of our time with now is trying to make things like trying to find partners internationally. And so we're hoping that now we've got a few different people starting that will, you know, like kind of like we did throughout North America, it'll just sort of start taking off. So that's our hopes. But yeah, that's what we're really trying to do. So I mean, like I can't promise it, but like we're buying cameras for partners internationally right now, for example, just to make certain that like, you know, like we want to make certain that you are a partner in this and you have the tools available to you if you potentially do not have them right now. So
oh, that's great. Yeah,
yeah.
I'm glad this person asked. Thank you for that question, because yeah, I think if there are other people like Josue too, he's saying that in Ecuador too, he can start thinking about projects there and stuff, and well, be in Guatemala, and maybe there are others here too that can.
Yeah, it would be
that
amazing.
Could join. Yeah, that's fun. I don't know how I've never asked you that, so I'm glad someone did. So I think that's it, John. Cool. No more questions.
Yeah, I mean, I would try to take some time here to convince somebody in Lincoln to join UWIN, but that's already been done. So I'm really excited to see what Andrew's actually going to be coming up with in terms of data and where species are located throughout your city. So
yeah.
Okay, well, thanks a lot, Mason. Great talk, and everyone for coming, and we'll hopefully see you all next week.
Yeah. Thank you all so much for the invite.
Thanks, Mason.